Hello and welcome back to the next session of microbes in human welfare. Now in the earlier part we came across microbes in household products, and in that part we came across various food products that we use in our day to day life. Now let's come back to the next part, which is regarding microbes in industrial products, or we also call it as microbes in industries. Now microbes in industries and microbes in household food products, the basic difference between these two is, in the industries we are going to produce them in large quantities. In the house we are going to produce food in small quantity, because the total number of people who are present in our homes, their number is less, production or cooking up of the food is also less, while in case of the industries it is a large scale. Okay, it is a commercial part, it is a commercial view. Okay, now in this, as we come across microbes in the industries, let's go to the first part, microbes in major industry, where we call it as a fermentation, alcoholic fermentation. So microbes in fermented beverages. Now fermented beverages are those alcoholic products which are produced by the industries in large quantity. Okay, now how these fermented beverages are produced? We have the larger stainless steel vessels, where we call them as reactors, bioreactors or fermenters. In these fermenters we are taking the mother liquor, where we call it as molasses. Molasses is the mother liquor that comes from all the sugar industries. Okay. Now to this mother liquor or molasses we are going to add yeast. The yeast that we are going to add here is what type of yeast? It is called as Saccharomyces cerevisiae. This is the yeast where we use in the preparation of the bread. The same type of yeast is also used here. Now this Saccharomyces cerevisiae, this is commonly called as a brewer's yeast. This is commonly called as a brewer's yeast. And its scientific name is Saccharomyces cerevisiae. If the yeast is used in bakery industries, we commonly call it as baker's yeast. If the same yeast is used in brewery industries, brewery is nothing but preparation or synthesis of beverages. That's why we call it as a breweries. Okay. Now in this brewery industries, we are going to produce the products in large quantity. By the process of fermentation, we are going to produce two different types of alcoholic products, where we call them as a distillation or distilled alcoholic products and non-distilled alcoholic products. Okay. Now by the fermentation process, we are going to produce, one is a non-distillation alcoholic products, and second one is the distillation alcoholic products. Non-distillation, where the concentration of the alcohol is very less. Now the non-distillation alcoholic products, here we have certain examples like beer and wine. These are the two alcoholic products which are produced by non-distillation process. Through the distillation, the alcohol concentration goes on increasing, and we are making it little bit more concentrated from the previous concentrated value. Now in case of this distillation, we have various different products. Apart from these two, beer and wine, the rest of the brands which you know comes under this category. Then what are those brands? So it comes like brandy, whiskey, rum, gin, vodka, etc. Okay, so all these are various different types of distilled alcoholic products. Distillation is nothing but it is a process where the concentration of alcohol goes on increasing by the removal of water. Okay. Then non-distillation, here the alcoholic percentage is less. Now yeast undergoes poisoning themselves at a alcoholic percentage of around 12 to 13%. That's why beyond that the alcohol percentage cannot be increased. We have to increase artificially through the industries by the process of distillation. And non-distillation, they are directly, these alcoholic products are obtained by the fermentation of yeast. Now out of all the brands, the least alcohol concentration is present in the beer, okay, which measures around 5 to 7% also. Highest alcohol percentage is in vodka, nearly around 80%. Okay. Then apart from that, we have one more type of alcoholic product where we call it as a rectified spirit. Rectified spirit is it is around 95% alcohol, and absolute alcohol this is 100% alcohol. Okay. So these two are the additional parts. Rectified spirit, it is around 90 to 95% of alcohol. Absolute alcohol is 100% alcohol, highly concentrated. What will happen if this high concentration, if a person consumes this rectified spirit, he will die on the spot, because it is too concentrated, right? That's why this rectified spirit, it is used in the spirit lamp, burning up of the lamp in the laboratories, spirit lamp. It is the rectified spirit only, right? And this rectified spirit, it forms the raw material for all the alcoholic products, okay, where the alcoholic concentration will be maintained properly. Now in the earlier days also we were having an alcoholic product where we used to call it as toddy. Toddy is nothing but it is the fermented sap of palm plant. That was the crude method, old method. Okay, now we have the modern method of production of alcohol by the industries in large quantity, and we are going to produce it. We are producing it. The highest sale marketing is from these alcoholic products only. Okay. So this is regarding the part of the first one, microbes in industries. That means in the industries we are using these microorganisms for our benefit, so that we can produce various types of products in different concentrations, in different grades. Okay. So this is the first part, fermented beverages. So with this, let's go to the second part. Second part is antibiotics. Antibiotics are also produced by the industries in large concentration. As the name says, antibiotics, anti means against bios means life, against life. These are the chemicals, substances which are produced against the life. Whose life? Our life? No, against the life of microorganisms, against the life of pathogens which causes disease in the humans. Okay. Now antibiotics are the chemical substances which are produced against the microorganisms or pathogens. Let us more accurately call it as pathogens. To kill the pathogens we are going to use this. Okay. Now this antibiotic, the first antibiotic that was produced was penicillin. The first antibiotic produced is penicillin, and this was produced from a fungus, and the name of the fungus is Penicillium notatum. It is called as a Penicillium notatum. This is the name of the fungus from which the first antibiotic penicillin was produced, and this penicillin, it was discovered by Alexander Fleming. Okay, This was discovered by Alexander Fleming. Alexander Fleming accidentally discovered penicillin. He did not purposely discover penicillin. He was doing another experiment. ಅವನ ಎಕ್ಸ್ಪೆರಿಮೆಂಟೇ ಬೇರೆ, ಆಕ್ಸಿಡೆಂಟಲಿ ಸಿಕ್ಕಿರುವಂಥದ್ದು ಪೆನ್ಸಿಲಿನ್ ಅಂತ ಅನ್ನುವಂಥದ್ದು. ಓಕೆ, ಒಮ್ಮೆಗೆಲ್ಲ ಸಿಕ್ಕಿಲ್ಲ. ವಾಟ್ ವಾಸ್ ಹೀಸ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್? ಹಿ ವಾಸ್ ಕ್ಯಾರಿಂಗ್ ಔಟ್ ಹೀಸ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್. ಹಾಗಿದ್ರೆ ಅವನು ಏನು ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಮಾಡ್ತಾ ಇದ್ದ? ಹಿ ವಾಸ್ ಕ್ಯಾರಿಂಗ್ ಔಟ್ ಹೀಸ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಆನ್ as he was carrying out his experiment on a bacteria actually, and that name of the bacteria is a Staphylococcus. Look at this, the name of the bacteria that was selected by him was Staphylococcus. In the laboratory, in a culture plate, ಒಂದು ಕಲ್ಚರ್ ಪ್ಲೇಟ್ ಒಳಗಡೆ ಈ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಮಾಡ್ತಾ ಇದ್ದ. ಈ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಮಾಡೋ ಟೈಮ್ ಒಳಗಡೆ ಏನಾಯಿತು? So what happened was, in the glass plate, his bacteria were here. On the surrounding, ಇದರ ಸುತ್ತಮುತ್ತಲೇ ಮೋಲ್ಡ್ ಬೆಳೀತಾ ಇತ್ತು. ಮೋಲ್ಡ್ ಅಂತಂದ್ರೆ ಫಂಗಸ್. That mold was not allowing his Staphylococcus bacteria to multiply. ಅವನ ಲ್ಯಾಬ್ರೋಟರಿ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಇದು. ಯಾವಾಗ ಈ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಸ್ಟಾಫಿಲೋಕೋಕಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಾನ ಇಲ್ಲಿ ಇರುವಂಥ ಕೆಲವೊಂದು ಸಬ್ಸ್ಟೆನ್ಸಸ್ ಅದನ್ನು ಮಲ್ಟಿಪ್ಲೈ ಮಾಡಲಿಕ್ಕೆ ಬಿಡಲಿಲ್ಲ, ಆವಾಗ ಅಲೆಕ್ಸಾಂಡರ್ ಫ್ಲೆಮಿಂಗ್ ಇದಕ್ಕೆ ಏನಂತ ಕರೆದ? ದೇ ಆರ್ ಹಿ ಕಾಲ್ಡ್ ಇಟ್ ಆಸ್ ಆ್ಯಂಟಿಬಯೋಸ್ ಅಂತ ಕರೆದ. ಆ್ಯಂಟಿ ಅಂದರೆ ಅಗೇನ್ಸ್ಟ್, ಬಯೋಸ್ ಮೀನ್ಸ್ ಲೈಫ್. ದಿಸ್ ವಾಸ್ ಅ ಸಬ್ಸ್ಟೆನ್ಸ್ ವಿಚ್ ವಾಸ್ ಪ್ರೊಡ್ಯೂಸ್ಡ್ ಅಗೇನ್ಸ್ಟ್ ದ ಲೈಫ್ ಆಫ್ ಹೀಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ. ಅದಕ್ಕಾಗಿ ಅದಕ್ಕೆ ಆ್ಯಂಟಿಬಯೋಸ್ ಅಂತ ಕೊಡ್ತಾನೆ. ಅದು ಮುಂದೆ ಹೋಗ್ತಾ ಅದು ಪೆನ್ಸಿಲಿನ್ ಅಂತ ಅನ್ನುವಂಥದ್ದು ಗೊತ್ತಾಗ್ತದೆ. ಫಂಗಸ್ ಪೆನ್ಸಿಲಿನ್ ನೋಟಾಟಮ್, ಅದರಿಂದ ಪೆನ್ಸಿಲಿನ್ ಅನ್ನುವಂಥದ್ದು ಬಂತು. This was the discovery. Okay, then Alexander Fleming, he just discovered this fungus. Then later on, its a therapeutical value, its medicinal properties ಅಂತ ಕರಿತೀವಿ. Its medicinal properties were identified by Einstein and Howard Florey. Its a therapeutical value, its medicinal value, its medicinal property was shown by Einstein, two of his friends Einstein and Howard Florey. Because of this, what happened? They all together, when they discovered this penicillin injection, this penicillin injection was widely used to the soldiers who were wounded in World War II of US. In America, American soldiers were wounded in the World War II in the year 1945. Because of this, when the penicillin was discovered, all of these three persons, Alexander Fleming, Chain and Florey, they have been awarded with the Nobel Prize. In the year 1945. Okay, they have been awarded with the Nobel Prize in the year 1945 due to the discovery of the penicillin, along with that by saving the life of thousands of soldiers. Okay, that's why today we have many different 1000 varieties of antibiotics like tetracycline, erythromycin, kanamycin, azithromycin. So different types of antibiotics are obtained from various sources. Mainly they are obtained from bacteria along with the fungus, but the first antibiotic which was obtained was from a fungus, and the name of the fungus is Penicillium notatum. Then what you need to remember here, ನೀವು ಏನು ನೆನಪಿಟ್ಕೋಬೇಕು? Name the first antibiotic that was discovered. It is penicillin. It was discovered from which microorganism? Penicillium notatum. Then who discovered it? Fleming. Alexander Fleming discovered it. Who gave the therapeutical value? Who gave the medicinal property, medicinal value for it? The medicinal value, it was mainly given by Ernest Chain, along with that Howard Florey. All of these three, Fleming, Chain, Florey, have been awarded with the Nobel Prize in the year 1945 by discovering the penicillin. Along with that, the penicillin saved life of many wounded soldiers in World War II. Okay, remember this one, right? So this is regarding the part of microbes in industries. In that, fermented beverages, and second one is antibiotics. Okay, now let's go to the last heading of this main part, last heading of the industrial part. It is regarding the chemicals, enzymes and bioactive molecules. Okay, so let us look at the next part. Next one is chemicals, enzymes and other bioactive molecules. Okay, out of these, let's come to the first heading. First heading is the chemicals. In this chemical, let us take the first one. I'll just go into an serial order, sequential order. The chemicals are ABCl chemicals. First one is acetic acid. Second one, butyric acid. Third one, citric acid. And fourth one, lactic acid. Now these are the four chemicals which are produced from different microorganisms. Acetic acid is discovered from Acetobacter aceti. Butyric acid is discovered from Clostridium butylicum. Citric acid is discovered from Aspergillus niger, and lactic acid is discovered from Lactobacillus or LAB. LAB stands for lactic acid bacteria. Okay. Now how to remember them? Because you need to remember chemicals, you need to remember enzymes and other bioactive molecules also. So let me call it as ABCl chemicals. Okay, so what is this? A for apple, alla. Now acetic acid and karitivi. B for ball, no butyric acid. C for cat. Here we have citric acid. So let me call it as a ABCl chemical. First term togondini. A, acetic acid, butyric acid, citric acid, lactic acid. You have only these four in your NCERT. So let us study only these four. Let us remember only these four. Okay. So how to remember them? Acetic acid is produced from Acetobacter aceti. Other naming malane irtada. Acetobacter aceti. Then butyric acid, Clostridium butylicum. Butylicum, butyric acid. Then citric acid. This is a little bit different. This is a fungus, Aspergillus niger. Lactic acid, you know it, Lactobacillus or lactic acid bacteria, LAB. Okay. Now, these are produced in the industries in large quantity, because all these are the chemicals, and these chemicals we are using in our lab. Not only in our lab, it is used in the degree colleges also, it is used in the PG colleges also, it is used by the research persons in the laboratories. So, throughout the globe, the chemicals are used, and hence they have to be produced in large quantity. They are produced in large quantity and they are transported in large quantity also. Okay. This is how, with the help of these microbes, we are going to produce these products. Okay. So these are the microbes from which the products are produced. These are the products, these are the microbes, because our title says microbes in human welfare. So how we are using them in our day to day life. These are chemicals. Let's go to the enzymes. Now in this we have the first one, proteases and pectinase. second lipase, third streptokinase. Okay. Now these are the enzymes which are used in industries to produce the products also. Protease and pectinase, they are used in clarification of fruit juices. In bottle juice products. Okay. Now protease and pectinase is an enzyme which is used in industries for the clarification of the fruit juice. Now for example, I will tell you what is this. In our home, we prepare orange juice. When we prepare orange juice, we will take the orange, we will peel out its skin, then put it in a mixer jar, then crush it properly, mix it properly, grind it, and later on filter it, then take it in a glass. But the solution or this juice what you take in the glass, it will not be a clear solution. But the orange juice that you bring from the market, where you carry the bottle, there the solution is very clear. This is because that turbidity is removed by these enzymes, protease and pectinase. That's why we find the clear fruit solution in case of the industrial packed bottles, and in our homes, homemade fruit juice, it will not be a clear solution because we are not using these enzymes in our homes, while industries will use them for packing up of this bottle juice products. Okay. So protease and pectinase are the enzymes which are used in clarification, means clearing the fruit juice so that the fruit juice appears a very clear solution. Okay. Lipase is an enzyme which is widely used to remove oil stains. So it is used to remove oil stains from the clothes. Suppose while eating the food, if any food particle falls on the shirt, okay, then it will be, if it is oily, then ultimately the oil cannot be removed so easily with water. For that, this lipase is an enzyme that is widely used in all the detergents, and it is also used by laundromans. Okay, it is widely used in laundries by laundromans. Okay, so this lipase enzyme is present in washing powder Nirma, Surf Excel, Ariel, in all the detergents. We have this lipase enzyme that is going to remove oil stains. Remember this one. Name the enzyme that is responsible for the removal of the oil stains. It is a lipase. Lipase removes oil stains from the clothes. Okay. Then let us go to the last one, streptokinase. Streptokinase is an enzyme that is obtained from the bacteria Streptococcus. It is obtained from Streptococcus. Okay. Now, this Streptococcus will produce an enzyme streptokinase, and this streptokinase enzyme is used as a clot buster. So, it is used as clot buster. This is the most important term that will be asked for an objective, even for your NEET, not only for the objective part of your theory, but for the NEET part also. Which enzyme is used as a clot buster? Remember, it is the streptokinase enzyme. Okay. The streptokinase is an enzyme that is used as a clot buster. What is this clot buster? It is used in MI patients. In myocardial infarction. It is a zoological medical term where we commonly call it as heart attack. In this persons, what happens, suppose if this is a blood vessel, if there is any clot here, ಇಲ್ಲಿ ಏನಾದರೂ ಕೊಲೆಸ್ಟ್ರಾಲ್ ಡಿಪಾಸಿಷನ್ ಆಗಿತ್ತು, ಕ್ಲಾಟಿಂಗ್ ಆಗಿತ್ತು ಅಂತಂದರೆ ದಿಸ್ ಈಸ್ ಎ ಇಂಜೆಕ್ಷನ್, ದಿಸ್ ಎ ಇಂಜೆಕ್ಷನ್ ವಿಚ್ ಇಸ್ ಗಿವನ್ ಬೈ ದ ಡಾಕ್ಟರ್ಸ್. ಓಕೆ, ಹಾರ್ಟ್ ವಾಲ್ ಮೇಲ್ಗಡೆ ಬ್ಲಡ್ ವೆಸಲ್ಸ್ ಇರ್ತಾವೆ, ಆ ಬ್ಲಡ್ ವೆಸಲ್ಸ್ ಕ್ಲಾಟಿಂಗ್ ಆಗಿತ್ತು ಅಂತಂದ್ರೆ ದಿಸ್ ಇಂಜೆಕ್ಷನ್ ಇಸ್ ಗಿವನ್. It is a clot, this is a clot, the injection will go and it will burst this, it will burst madam, it will burst that clot so that the blood can flow to other parts of the heart tissues. Okay, otherwise what happens, it leads to heart attack and where we call it as myocardial infarction. That's why such type of patients are called as myocardial patients. MI wards and irthav hospital ಒಳಗಡೆ, they are for this heart attack patients only. Okay, this is the streptokinase enzyme. This is regarding the part of the enzymes. Let's go to the last heading now, bioactive molecules. Now in the bioactive molecules, we are going to discuss only two bioactive molecules. First one is cyclosporine A. Then second one is statin. Cyclosporine A is produced from a microbe. It is produced from a microbe called as a Trichoderma polysporum. It is produced from Trichoderma polysporum. Statin is produced from Monascus purpurus. Both are fungal derivative only. Cyclosporine A is produced from, it is in the form of injection only. Statin is also injection, both are injections. Trichoderma polysporum is a fungus from which this cyclosporine A is produced, and statin is produced from Monascus purpurus. Then what are their function? Now this cyclosporine A, it is used as immunosuppressive agent. Immunosuppressive agent during organ transplantation. During organ transplantation, this is most important. Again, it is an objective part, which is used as a immunosuppressive agent. It is cyclosporine A, produced from Trichoderma polysporum. It is used in the form of injection. Now look at this. During organ transplantation, like kidney transplantation, suppose the kidney has to be replaced. If the kidney has to be replaced, then our immune system won't retain even a small microorganism, then how can it retain a larger kidney? That's why doctor will give an injection. The name of that injection is cyclosporine A, so that our body gets adjusted to this organ. Okay, during kidney transplantation, heart transplantation, ಅದಕ್ಕಾಗೆ ಇಂಥ ಟ್ರಾನ್ಸ್ಪ್ಲಾಂಟೇಷನ್ ಆದಾಗ ಡಾಕ್ಟರ್ಸ್ ಯೂಶ್ವಲಿ 2 to 3 months ಎಲ್ಲೂ ಹೊರಗಡೆ ಹೋಗಬೇಡ ಅಂತ ಅಂದಿರ್ತಾರೆ. Because your immune system will be so weak that you can get any kind of diseases, right? Because we are present in potentially harmful environmental conditions. Okay, this is cyclosporine A. Then statin, it is used to lower, it is used as blood cholesterol lowering agent. Look at this, this is also most important. Now this is used as a blood cholesterol lowering agent. Which one? Statin. Suppose if the cholesterol level goes on increasing in our blood, what will happen? It leads to blockage of the blood vessels, leading to heart attack and death. As a result, as the age proceeds, almost doctors will suggest for the aged persons as well as for the MI patients that don't consume more cholesterol. This is because if cholesterol concentration increases, chance of heart attack is high, risk of heart attack is high. As a result, the cholesterol has to be kept under control. Now statin, it lowers from the blood. If the cholesterol content is high, the cholesterol content is lowered by this type of injection. That is why blood cholesterol lowering agent is statin. Immunosuppressive agent is cyclosporine A. Okay. So this is the chemicals, 4 chemicals, 4 enzymes, 1, 2, 3 and 4 enzymes and 2 bioactive molecules. So this completes the second part of the topic. Then the later parts of the topic we shall continue in the coming class. Thank you. Have a nice day.